Hello, everyone, and welcome to this audio podcast. This audio podcast overview as part of a research and report. This audio podcast an overview as part of a Labostix report, and this audio podcast as part of a ZtormLabostix production. Today, looking at works in regard to pathology approaches to easing kidney disease burden. Pathology approaches to easing kidney disease burden, and where this works was formed a sort of chapter contribution to a Springer pathology or biomedical book chapter contribution in around 2023, so in this overview there will be a look at some of the bases of this of the collaborative works. This audio podcast again also is a part of a ZtormLabostix Collaborative. So to begin with, some contents which includes and where in this overview we look at introduction, looking at biomedical classification, looking at aims and objectives, looking at complexity in context of complexity in around the sort of basis of viruses, and looking at also the sort of renal and COVID-19 correlation, and then looking at the taxonomy and overview of coronaviruses, and then biomedical analysis of COVID-19, and then laboratory practices, looking at laboratory practices, and then conclusions, and then a sort of a reference or citation towards the end of this overview. So to begin with, some introduction. So the coronaviruses can affect multiple organs in humans. For example, the kidneys, increasing the mortality, especially in those who are immunosuppressed and or immunocompromised. Moreover, the lack of treatment forces the biomedical and pharmaceutical industries to seek approaches to offset healthcare crises. And where there is a lack of effective treatment for SARS-CoV and SARS-CoV-2, a more indirect approach should be considered to lower probable risk and on a set of disease amongst immunocompromised and immunosuppressed individuals and recipients. So the research surrounding COVID-19 has generally been progressive, and it is important to reduce disease mortality at epidemiological and population level. However, the understanding of structure and pathology of coronaviruses, especially surrounding the host pathogen communication and visualization, requires insight. So looking at sort of the biomedical classification, so the coronaviruses are actually a more recent discovery than that of the adenoviruses. Coronaviruses, just to sort of iterate, coronaviruses stem from the adenovirus, and where the recent discovery that where in around recent discovery that fall under the same category, and where retrospectively there has been more there have been more taxonomy investigated. The first coronavirus strain, the B814, had been and was discovered in around the mid 1960s by investigators and investigative scientists Tyrell and Bino, where biomedical specimens were collected from the respiratory tract of a recipient with a common cold. Subsequently, samples were then linked directly to human volunteers to develop the disease and evidencing the presence of infectious agents. Thereafter, investigators Hammer and Procknell were then able to grow isolated virus strains, including the B814 and 229E, which improved early biomedical visualization in context of extraordinary viruses. Now, in around the sort of aims and objectives, so this overview seeks to highlight the causes and potential mechanisms or mechanistics during severe acute respiratory syndrome or SARS infection CoV-2, the sort of renal interaction and the effects of acute kidney injury or AKI. And the overview also looks to provide a review of pathology and analysis of approaches to easing kidney disease burden by exploring or prompting sort of an awareness of host pathogen communication, providing a biomedical visualization perspective. So looking at sort of some of the complexity. So based on taxonomy and pathological features, a team of 8 virologists initially decided to classify coronaviruses as to classify sort of coronaviruses, and due to specific and also particular characteristics and appearance. Ongoing research and microscopy advances surrounding coronaviruses have provided greater knowledge of viral structure and function to discover therapeutic or pharmacotherapy possibilities around unpredictable outbreaks. Advanced microscopy and visualization have been important throughout practices and updates surrounding the pandemic. So in around the renal and COVID-19 correlation, so it had been highlighted or even postulated that the first pandemic caused by coronavirus, SARS-CoV, which occurred in around 2002-2003 in Asia, wherein just over 8,000 recipients were evaluated for the virus, and as a result, 700, just over 700 or 774 deaths had been documented. So, despite common acute respiratory failure caused by diffuse alveolar damage, multi-organ failure was also noticeable when there were cases of severe diarrhea, hepatic failure, and AKI. An investigator from Hong Kong, after evaluation, highlighted that 536 SARS of cases, around 6.7% of patients developed AKI. Moreover, the mortality amongst the cohort was in around almost 92%, 91.7%, which is and was significantly higher than recipients without any symptoms of AKI, which was in around just under 9%, 8.8%. Now looking at sort of the taxonomy and overview of coronaviruses, and in this on this sort of slide, if you like, there is a schematic or a schema, and to inform in around this and around this, the schema can be divided into three columns. So on the far left you have, on the far left of the schema, you have the order. You have the order of the particular general or the family the general or the family of the kind of the sort of the viruses. And then and then in the middle, in the middle, in sort of the middle column, you have the lineage, and where the lineage is connected to the sort of genera, the family and the genera, of particular of the genus of the particular viruses. And then on the far right you have on the far right of the schema, you have how you have a sort of basis where the lineage, where particular lineage is connected to to specific forms of the coronaviruses. So for example, from the left, going from the left, you have, for example, you have the nidovirales, viruses. You have the coronaviride virus, and you have the coronavirinae viruses, coronavirinae, a family of sort of viruses. And then the genus then split up. They split up between the alpha coronaviruses, the beta coronaviruses, the gamma coronaviruses, and then the delta coronaviruses. And then the four genus sort of coronaviruses are linked to specific linked to specific lineage A, B, C, and D. A, B, C, and D. In context in context, and particularly in around the beta coronaviruses. In the genus around the alpha coronaviruses, you have alpha coronavirus 1, where you have HCOV 229E and HCOV NL63. And then there are several lineages of viruses that connect or are particular and specific to the beta coronaviruses. For example, you have under lineage A, you have murine coronaviruses. In the lineage B, you have SARS. You have SARS-CoV, and you have the MERS-CoV. And under lineage B, you have the SARS-CoV, and under lineage C, you have the MERS-CoV, in addition to BT-CoV, and HKU4, and BT-CoV, HKU5, and then under lineage D, you have BT-CoV, HKU9, and then under the gamma coronaviruses, you have the avian coronavirus, and under the Delta coronavirus, you have the BU-CoV HKU11. There are in around the taxonomy there in around the taxonomy there are publications in amongst in amongst literature in around virology, particularly that go into the sort of the sort of more particular more particulars around the alpha coronaviruses, beta coronaviruses, the gamma coronaviruses, and delta coronaviruses, and so this schema this schema just sort of provides a brief illustration a brief illustration or overview of the taxonomy and overview. So now looking at biomedical analysis of COVID-19. So a SARS-CoV-2 virion contains an unsegmented single-stranded positive sense RNA genome of around 30 kilobases, which encodes crucial viral protein used to create progeny viruses and the four essential structural proteins called the membrane, the envelope, and the nucleocapsid, and the spike. As far as airborne viruses are concerned, SARS-CoV-2 is transmitted between humans due to entering the viral particles to the upper respiratory tract. The viral entry occurs by the binding of the spike glycoproteins, which are heterotrimers present on the surface of coronaviruses, present on the surface of the coronaviruses, and where there is some bases in around the ACE angiotensin converting enzyme 2 receptor as well. To also iterate here that the spike glycoproteins where are those sort of glycoproteins which are sort of the spike formations that are in amongst various illustrations or schema in amongst published works, where both sort of adenoviruses and coronaviruses are concerned. The spike proteins are cleaved by host proteases into subunits. S1 subunit for receptor binding, and S2 subunit for membrane fusion. Microscopically, the infected cells are forced to translate proteins required for generating the virus's progeny. So to iterate here, the infected cells, or where infected cells are concerned, it is the glycoproteins or the spike glycoproteins that sort of force the sort of a transcription for the generating of the virus's progeny. So, in context of laboratory practices, or where literature has highlighted some bases in around the investigations of the sort of renal and the SARS, the renal and the SARS sort of affiliation in some additional investigations. is where most informative methodology to analysis disruption caused by SARS-CoV-2 is through the biopsy of renal tissue or kidney tissue, with the usage of light and electron microscopy. Samples overgo sort of structural investigation to identify injury within tubular, epithelial, and glomerular cells. The tubular, to iterate here, the tubular, epithelial, and glomerular cells are those cells are those cells or those particular parts parts of um, those particular parts of sort of renal anatomy, which are in some regard most sort of more vulnerable or more fragile, where injury, where tissue injury is concerned. And so linked to this is where podocytopathy has been identified, which in effect is a glomerular disorder, and is mostly observed within COVID-19 recipients, possibly caused by cytokine mediated podocyte injury. And microscopy evaluation has also identified in around and of kidneys in recipients with COVID-19, um, COVID-19 have provided a sort of an array of renal complications, where renal histopathology and pathophysiology complications have been identified in around some of the laboratory practices, some of the laboratory practices where COVID-19 COVID-19 and the renal correlation is concerned. And this is there is a requirement, therefore, for further research to understand sort of the data which could potentially provide clearer understanding of renal microscopy affected by COVID-19 in sort of in through sort of prospective investigations. So now looking at some of the conclusions. So there is perhaps an indirect advantage in implementing ACE2 or angiotensin converting enzyme to treatment to improve the immune response against infection. ACE2 may also offer appropriate strategies for the management of symptoms or symptomologies that relate SARS-CoV and SARS-CoV-2 in most immunocompromised or immunosuppressed recipients. However, what is still not known is whether ACE2 should be assigned at population level to totally prevent COVID-19 symptoms or symptomology. That notwithstanding, advances in laboratory practice allow implementation of pathology assays and techniques, including microscopy advances for visualization and analysis to preclude kidney disease burden. On this slide here, just an overview of the reference or the citation for this works, and this was works that was again formed a chapter contribution in a Springer publication, Advances in Experimental Medicine and Biology, Volume 15, Visualization in Teaching of Biomedical and Clinical Subjects, Anatomy, Advanced Microscopy, and Radiology 1406 or 1406, and again the the chapter publication can be accessed through online by searching the by searching databases such as PubMed or Medline. If not the full chapter publication, then the abstract to part of this overview is available through PubMed. Should be available through PubMed and Medline, or through Springer's Springer's sort of Springer's website or database. So that draws this overview to a close. This overview as part of a research and report. This overview as part of a Labostix report, and this audio and this podcast and this overview as part of a ZtormLabostix Productions.